everyone, and welcome to the Arthritis National Research Foundation's Researcher Spotlight webinar series. I'm Emily Boyd Stormoen. I'm the CEO of the ANRF, and on behalf of our Board of Directors, our Scientific Advisory Board, and our staff, I'd like to welcome you to the Spotlight on Rheumatoid Arthritis. We're so excited to have many of you joining us from all over the world. And I'd also like to recognize our sponsor for this webinar, AbbVie. Thank you for your support. Now, I'd like to welcome the moderator for our discussion, Dr. Elizabeth Mellins. Dr. Mellins is a professor of pediatrics at Stanford Medical Center and a member of the ANRF Scientific Advisory Board. Thank you, Dr. Mellins, for moderating this webinar today. Before we get started, there are a couple of logistical pieces of information that we wanted to share. First, the webinar will be recorded, and we'll have it available on our website and on our YouTube channel in the coming weeks. If you have any questions during the webinar, please type them into the Q&A. If we aren't able to get to yours, please email info@curearthritis.org after the webinar with your question. Now, let's get started with today's webinar and hear more from our ANRF scholars. Dr. Mellins, I'll turn it over to you.
Thanks very much, Emily. So I would just want to start by also welcoming you to what is a very exciting now yearly event sponsored by ANRF, where we spotlight active young researchers who are currently supported by ANRF, and we do it in a topically oriented way so that each of the talks in certain ways synergizes with the other talks. I also just want to add about myself that I was an ANRF recipient, but I won't tell you how many years ago. So we will start with our first speaker, who is Susan Maclauchlan, PhD. She's an instructor in medicine at Brigham and Women's at Harvard Medical School. And her topic, which will be, I think, of great interest to anyone who has rheumatoid arthritis or knows someone with rheumatoid arthritis, is seeking new mechanisms linking rheumatoid arthritis to cardiovascular disease. Thank you, Susan.
Thank you for that introduction. I'm really honored to be here today and really honored at the idea that the future is looking bright if I can look forward to one day moderating such a panel and saying I was a recipient of the ANRF. So thank you for that. Have I entered presenter mode? Do my slides look good?
They look
fantastic. Fantastic. Well, my ANRF was about trying to understand cardiovascular disease in the context of rheumatoid arthritis. And I'd really like to thank the ANRF, not only for selecting me, but also being a unique mechanism where someone who's traditionally an outsider to the rheumatology field could be welcomed in, encouraged to take on a bold study and to begin research in a slightly new field. So I wanted to start off by sort of highlighting some of the things that I've accomplished while on this ANRF, which I think you can read through yourself. But you know, one of the important things that for me personally has been helpful are these are studies that I'm actually hoping to leverage and convert into my own personal independent funding, which is called an R01 application, which will allow me to open up my own independent laboratory. So that's the next big career milestone for me, and the ANRF has given me protected time and resources to start to pursue those things. So I'm very grateful for that. Yeah, I think one of the things that is maybe not even well appreciated in people who study cardiovascular disease is that our patients with rheumatoid arthritis are at an increased risk of cardiovascular disease, and we really don't understand that very well at all. So when we, as someone who's trained in cardiovascular biology, we think about diabetes as being a major disease that we study, and diabetes doubles the risk of cardiovascular disease in that patient population. And we've learned a lot from studying diabetes. And I think that we can well argue that because the patient population in RA doesn't fit the traditional cardiovascular risk factors that we've so well understood in the general population, there's a clear unmet clinical need to understand why inflammation that targets the joint also leads to systemic inflammation that's impacting our blood vessels and our hearts. And this is really an important factor, I think, because as I alluded to, those traditional risk factors aren't actually describing the patient with rheumatoid arthritis that goes on to have cardiovascular disease in many instances. So there's room for improvement in understanding why patients with cardiovascular disease and with rheumatoid arthritis end up with cardiovascular disease. And another important factor that I think leads to the prescience of these kind of questions is that our standard DMARD therapy, so the standard therapy to limit the inflammation at the joint, is actually not being very effective at decreasing the risk to cardiovascular disease. So even these transformative medicines that have had such a great impact in improving the joint, stopping the joint inflammation, we have more to do for our RA patient population. So the work that I've been working on the ANRF grant itself, I thought I'd take today to actually take a step back and talk about how a study I did in a traditional cardiovascular space inspired me to then make this switch to try and leverage lessons that we'd learned in that space to the RA patient population and hopefully understand that better. So as I summarized, we're trying to think about novel risk factors that might link rheumatoid arthritis and cardiovascular disease. So I'd actually like to tell you now about work that I've been involved in that is highlighting a new cardiovascular risk factor in people. And that's called clonal hematopoiesis, which you can sort of think of as attack of the clones. And so what we've discovered is that the genome that we're born with over time as cells divide actually changes. And there's lots of ways that the body has to try and prevent this from happening. So it's not a very frequent event, but occasionally mutations that happen after birth can occur in tissues. So I'm showing you here a mosaic. And if you think about each of those tiles as a cell, and those blue cells as your original genome, you might have a cell that develops a mutation that's then able to proliferate. And so tissues become mosaics of themselves, of that original genome. And when this happens in the bone marrow, if it happens to the progenitor cells that give rise to our immune system, you can then detect it in blood with a blood draw and doing very deep sequencing of the genome in the immune cells. So this was actually done by Ben Ebert's group, who's over at the Dana-Farber. And they asked a couple of simple questions, which is how often does this happen and what's the consequence? So in that paper, which was published a few years ago in the New England Journal of Medicine, when they looked at how often does this clonal hematopoiesis event happen in healthy people, otherwise healthy people, what they found was as they looked over time with age, the frequency increases. As one might expect, there's more proliferative events and more opportunities for these mutations to occur. And then unfortunately for people, what they found was that patients that had these clonal hematopoiesis mutations, which is shown here in red, if they ask how long does it take until these patients die, unfortunately, the patients with the mutation have a lower survival overall compared to patients without a mutation. And a few of these people, I've told you there's mutations. One of the things you might think is, well, they're getting cancers. And indeed, a few of them did. But when Ben Ebert's group went and asked, what is causing mortality in that red line in those patients with clonal hematopoiesis, what came out was that, in fact, it was cardiovascular disease that was killing these people. So I'm showing you here in red coronary heart disease and ischemic stroke, and looking at the probability of an occurrence over time. It was more common in these people with these singular mutations, which was completely unexpected to us. So as a mouse biologist, we sort of said, well, we can add to this literature by doing mouse studies, which can take us, instead of looking at people generally, we can, in a very tightly controlled fashion, say, can we model this? And in only changing one thing, ask, is that occurrence sufficient to cause disease? Which is what we did. We looked at the genes that are most commonly affected by clonal hematopoiesis. And so we chose TET2 because a lot of the mutations, it's a very frequent mutation that often leads to loss of function. So we were able to leverage the fact that a TET2 knockout mouse already existed. And we were also able to utilize the fact that we have genetically altered mice that are prone to atherosclerosis. So what we did is we took this atherosclerosis prone mouse, we gave them bone marrow transplants, so removed the hematopoietic compartment, and either did this by irradiation, and then either gave them back wild type bone marrow, so a totally normal mouse, or gave them back a combination of wild type and a few of these hematopoietic progenitor cells that were clonal hematopoiesis mutants. And with genetic tricks, we were able to monitor the engraftment of those clonal hematopoiesis clones and their proliferation and expansion into the blood. So not only do they survive and expand, but they do quite well. And over time, there's more and more of them. And then to get these mice to develop atherosclerosis, we fed them a high-fat, high cholesterol diet, which is also known as the Western diet. Then to look at atherosclerosis development after a period of time, we actually looked at the atherosclerotic lesions in the heart in these mice. Those are designated here by the dashed lines around these plaques. And what you're looking at is the tricuspid valves of a mouse. So you're looking at a section through the heart this direction, down the aorta into the heart. And when we quantified how big these plaques were, in these clonal hematopoiesis mice on the right, shown in the black dots, we actually found that these mice were developing more atherosclerosis, which indicated to us that indeed clonal hematopoiesis has a role in cardiovascular disease progression. And so we were trying to model, why might this be? So we asked whether these TET2 knockout cells were producing more cytokines, which are pro-inflammatory and disease-causing molecules. And when we stimulated them with a pro-inflammatory agent, in the black bars, you can see that they produce more of several key cytokines, which I noticed were also many of our drug targets for the disease-modifying anti-rheumatic drugs that are the current standard of therapy for RA patients. And so this made me question whether or not there might be a connection between these processes and RA and broadly. And is this an opportunity then to sort of leverage what we've learned within the atherosclerosis studies and ask whether or not these are lessons that we can apply to rheumatoid arthritis more broadly? And so that's what I brought to the ANRF, and had several ideas about how to do that. Asking whether or not RA might worsen clonal hematopoiesis, and if clonal hematopoiesis is worsening atherosclerosis in the context of rheumatoid arthritis. And so that's what I've been working on, and it's not published yet, so I won't tell you more about that, but I hope to be invited back to tell you more about it in the future. And before I turn the floor over, I do want to highlight that this has been work that's been only possible because of great collaborators, many of whom I've listed here. And the people that I work around have been really great in teaching me more about rheumatoid arthritis, as well as the collaborators that we worked together to establish the clonal hematopoiesis model. So I would happily take any questions. And yes.
Emily, are there any questions yet?
No questions yet, but if you do have a question, please feel free to type it into the Q&A, and we'll make sure to get that to our speakers today.
So I'll start with a question then. So
fantastic.
Susan, so you started with the observation that the risk factors that we knew about for obesity or type 2 diabetes were not seeming to be the risk factors that would be driving an increased risk of cardiovascular disease in RA. So now we see a new cardiovascular risk factor in RA, but does it go the other way? Is there a synergistic additional hit that people with obesity or people with type 2 diabetes have, perhaps as they age, whatever? Does clonal hematopoiesis play a role in these other settings? And if not, are there other diseases where you're hypothesizing clonal hematopoiesis may be playing a cardiovascular inducing role?
I see. So you're asking whether or not the cardiovascular disease in other contexts, such as obesity, is driven by clonal hematopoiesis.
Or is
Yeah.
it whether clonal hematopoiesis is making a contribution? I'm sort of flipping your first observation that the risk factors were not the same and sort of flipping it on its head. So now you've got a new risk factor in RA. Does it have any impact in these other diseases where cardiovascular risk is a problem?
Yeah, and I think I'm one of many people who are thinking about this. So the first line that we've all been going through is clonal hematopoiesis impacting inflammatory diseases generally? And there's been quite a bit of research into that, and most of the places that we've looked, they have been. We've also been involved in a couple of other cardiovascular models, obesity itself, for instance. But in terms of modeling these multiple diseases, I'd actually say that's one of the places where our animal models fail, and suddenly they are no longer great at capturing all aspects of all of these diseases. And so, trying to merge these two diseases, RA and cardiovascular disease, in this in the case of my ANRF, was something that I feel is sort of an unmet need in terms of our experimental approach, and there isn't a good one out there. And so I'm hoping that with the work that we're doing, we can get closer to addressing that, because it's something we should be thinking about more. There's a lot more coming out of the epidemiological studies to speak to that, for sure.
Emily?
I think that's our questions for now, but we can also take questions through the webinar and go back to the presenters as well, if that works.
Okay, I would, I was just going to ask one more, if that's okay.
Please.
Okay. Because, it looked appealing. Appealing's not really the right word. Reassuring that the cytokines that were being stimulated were cytokines that we already had drugs to hit, and we were already hit. But the issue is we were already hitting them. So
absolutely.
So what does that mean? Kind of obviously.
Yeah, I think it's a great question, a really essential one, because you might say, maybe you don't expect to see so much clonal hematopoiesis because this is a population that we're already addressing these and targeting these things in. And, is it a way that maybe we can stop it? Because, if you can stop that process, that might help quite a bit. So those are things that I pay attention to in the literature, because I think that has a broad applicability to the patients and to understanding these diseases broadly.
Right. Yeah, I mean, I guess I think about broader or no, I mean higher numbers. Would you see a difference in the frequency of clonal hematopoiesis in a set of RA patients who were only treated with NSAIDs or something like that, compared to those who were on anti-IL-1 or anti-IL-6? So
those are definitely questions that we're looking into addressing and working with Ben Ebert to hopefully address those kinds of things. So would you like to review my grant? That's not how that works, unfortunately.
Unfortunately. Okay. Well, we will then move on. Thank you very much for a really stimulating presentation. It was great. Okay, so Renuka. Renuka Nayak is an MD PhD, assistant professor of medicine up the road from me at UCSF School of Medicine, living, I have to say, in one of the more beautiful parts of the world. And her title is, I also have to say, this title really made me smile. I now have a 19-month-old grandson, and this title, without in any way being disrespectful, reminded me of the title of one of the books I've read to him. Okay, it is Say Hello to My Little Friend, the Gut Microbiome in the Treatment of Rheumatoid Arthritis. Take it away.
Thank you so much for that introduction. All right, let me see. Is this coming across properly, or do I need to swap the displays?
Well, I do see the stuff on the far left, so it's not a full screenshot.
How about this? Is that better?
Still see the things on the left. Along the bottom, do you have a place to click that shows a screen that would make this full screen? If not, it's not a huge deal. I mean, we can see everything, so I wouldn't. If this is what you should go for it.
Okay. Let me see if, yeah, I think this is what I have.
Okay, no problem.
Let me try one more thing. Hold on 1 second.
Okay.
How about this?
Perfect.
Okay, good. Nice. And I'm just going to change my. All right, here we go. Yes. So yeah, actually I'm so pleased to hear that you said this is from a child's book. This is also from like a very scary movie. And it's in reference to something that is supposed to be very powerful. And that's part of the reason I chose this title, because the microbiome, I think, is actually very powerful in shaping our health. And we're just now starting to kind of appreciate the ways in which the microbiome can affect not just health, but also treatment of disease, which is the focus of my research. And so, I think some of you may have heard about the microbiome in the news, but for those of you are not familiar, I usually like to just get our mindset into the idea of what is the microbiome. And it usually refers to a collection of microbes that includes bacteria, but also viruses and fungi and archaea. So it's all of these guys, as well as the genes that they're bringing with us, because that is like where all the action is happening. It's the genes that they're bringing with them that shapes our own health. And with respect to the microbiome, there's actually different kinds of microbiome and microbiomes. And as a physician scientist, the one that I'm most interested in is the human microbiome, and in particular the gut microbiome. But there's microbes actually all over us, on our face, on our skin, and our ears, and the collection of those microbes that exist in each of these locations actually differs quite a bit. But other researchers are also interested in looking at other types of microbiomes, like the ocean microbiome or the soil microbiome. And what we've come to appreciate is that these microbes living in these different spaces actually can shape their environments in significant ways. So one sort of piece of evidence for this, or maybe some of you might be thinking, why is the microbiome important? And as a scientist, like what I like to ask is, well, what happens when you get rid of it? Is it important? And what we know from animal studies where we can actually get rid of a microbiome, we can't really do that with ourselves, with humans, but we can do this with animal models and rear them, like grow them up in these germ-free isolators, and then ask, well, how are these animals that don't have their native microbes on them, how do they behave, and how are they similar or different to a colonized mouse? And what we know is that these germ-free animals are actually nutritionally underdeveloped. They do not gain weight as quickly as their colonized counterparts. And that's because we rely on the microbes in our gut to help us digest the foods that we eat. And that's where all those genes that they're bringing with them come from. They have genes that we don't have that can do all of this enzymatic metabolic activities. But what we also know, and I think this is surprising actually to a lot of scientists, was that these germ-free mice have a stunted immune system, and that they're more susceptible to infections. And that is because the microbiome, our gut bacteria, also our skin bacteria, educate our immune system. So these microbes are interacting with our immune system. We used to think only the immune system was designed to combat microbes. What we're realizing is that there's this bi-directional interaction. And then I think this is also a very big surprise that germ-free animals actually exhibit some neurologic deficits. They behave differently than their colonized counterparts. They're anxious, they're hyperactive, and they don't seem to learn as well as mice that have all of their microbes with them. And so because these are really important functions that these microbes are conferring to their host, microbiome researchers like myself think of the microbiome as extending the capabilities of our own genome. And so we think of them as a second genome to our own genome. And if we understand more about these microbes and their genes, then we can actually develop therapies to target either the microbial genes or the microbial constituents and advance health in that way and advance treatment in that way. So everyone's microbiome is very personalized. It's almost like a fingerprint. The collection of microbes in my gut is going to be very different than the collection of microbes in your gut. And that's because the microbiome, the community of those microbes and the genes that they are bringing with them is shaped by a lot of different factors, by the method by which we were born, the kind of feeding that we got when we were little, the age that we're at, where we grew up, the drugs that we're taking, the diet that we're eating, the stresses that we experience all seem to impact the microbiome. And so given this, you could imagine that these microbes that are really playing an important role for us, they're interacting with our immune system. And I'm going to show you some data later that suggests that they're also interacting with the drugs that we give them. All this variation across us may explain some of the variation that we see in patients, why some patients respond to drugs and other people don't, why some people have side effects and others don't. And so that actually leads me to my next slide, which is that, when I first started in rheumatology, I was so sort of surprised to learn how variable patients responded to, how much variation there was in patient response. So I would see my rheumatoid arthritis patients in my clinic. I would give them the bad news that they have rheumatoid arthritis. And now almost every rheumatoid arthritis patient gets methotrexate. And what I would have to say to the patient is like, I don't know if this is going to work for you, but we're going to have to try it. And we have to give it like a good 3 to 6 months. And you might be in that 1/3 of patients that responds really well to this drug, or you might have like medium response, or it may not work for you at all. And, that's not a very satisfying conversation to have with a patient. I'm sure it's not satisfying from the patient end either, but that is what we saw, that there's a lot of variation. And so this variation in response to methotrexate has been actually the subject of a lot of different research endeavors. And investigators have looked at whether our own human genome contributes to this variation, whether factors such as smoking, diet, or the state of the immune system may contribute to this variation, or even the other medications that patients are taking, like the NSAIDs we heard about earlier, or IL-1 or TNF antagonists might be contributing. And so what I do think all of these are contributing to some of the variation, but we're starting to get some information that the microbiome contributes as well. And this phenomenon about the microbiome playing a role in treatment response is not just unique to rheumatoid arthritis, but exists in multiple clinical disciplines such as HIV prevention or cancer therapy. And so that was when I started my research looking as a rheumatology fellow, I really wanted to know, I saw these patients in my clinic, they were not responding the same way to methotrexate. People had looked at all these other things, but no one had looked at the microbiome. So I wanted to look at that. And so the first question that I asked was whether these microbes can act on methotrexate, and other drugs is also of interest to me, but I'm going to focus on methotrexate for today. And by, for example, metabolizing the drug with all the enzymes that they're bringing with them, could they affect drug pharmacology? Could they be metabolizing methotrexate into something that's not active? Because methotrexate is an oral drug, and it will encounter all of the microbes in your gut before it gets into your bloodstream. And so I'm going to show you one piece of data to support this idea that methotrexate is in fact metabolized by our gut microbes. And maybe that's why some of my patients are not responding to methotrexate. And if we knew that information ahead of time, that would be really useful to know before starting them on methotrexate. And so I'm going to show you some data to support this pathway. But in doing this research, I came across another unexpected finding, which is that when I took methotrexate and I incubated it with bacteria to look for metabolism, what I actually saw was that methotrexate was acting on the microbes and preventing their growth. And this was totally unexpected because methotrexate was designed to target human cells. And I saw that it was acting on bacteria. So this was like an off-target effect of methotrexate on bacteria. And then I realized that if bacteria can affect the immune system, maybe methotrexate is acting on the bacteria to affect the immune system. And so that's what the second pathway of research was for me, looking at how methotrexate affects the microbiome and how that might affect the host's immune system. And so for just this first pathway, I want to show you one piece of data, which reflects the experiments that I did looking at whether human gut microbes, like here's one of them, Clostridium aspergiforme, I incubated it with methotrexate and I looked across time. And this is kind of a special assay to look at a drug. And so methotrexate is this kind of big black peak here, and what happens to it over time. And you can see that peak is disappearing. with longer incubations with C. aspergiforme. And there's a new peak that I highlighted here in yellow that's coming up. And so this is pretty good evidence that methotrexate is being metabolized by C. aspergiforme. And so I did this kind of assay or test with 45 different human gut organisms, human gut species that are very diverse with respect to all the different categories that they come from. And all the ones that are highlighted here in red are the microbes that I saw can metabolize methotrexate. And these are very common in the human gut. And so there's actually quite a few human gut microbes that live in us and in my RA patients that have the capacity to metabolize methotrexate. But that was all like in a petri dish or in a test tube. I actually wanted to know also what's happening in my patients. And so, we have had the great privilege here of collecting cohorts of rheumatoid arthritis patients and following them over time to understand whether they went on to be methotrexate responders or non-responders. And this is also a collaboration with my co-investigator, Jose Scher at NYU. And what we saw was that when we looked at the microbiomes of RA patients before and after methotrexate, we were looking for whether there's any changes that are induced by methotrexate. When we looked at kind of all of them as a whole, we didn't see consistent changes. But when we separated them out into responders and non-responders, we saw that almost all the responders showed a decrease in this category of bacteria called Bacteroidetes, and that was like a significant shift compared to people who were non-responders. And this really suggests then that how the microbiome is responding to the methotrexate is associated with clinical response. And that was really exciting to see this connection between what's happening to the microbiome and how the patients are responding to methotrexate. And so I don't have time to those two lines of investigation have really flourished in the time that I've been in the lab and have led to these two publications that I'm highlighting here. And kind of the bottom line of all this research has been that methotrexate is metabolized by human gut microbes, and also methotrexate affects bacterial species, and that when it acts on microbes, it actually reduces the inflammation that's caused by those microbes. So, I think methotrexate not only acts on the host, but also acts on the microbes to reduce inflammation. That's a really, exciting finding. And we're trying to understand what all the mechanisms are in these two kind of interacting or interrelated findings that microbes act on methotrexate. I kind of didn't have time to show you, but that this metabolism is associated with how my patients are responding to methotrexate, and also that methotrexate acts on the microbiome, and in doing so, it reduces post-inflammation. And so the ANRF project has supported me in looking at, well, what are these new metabolites that are being produced by the microbiota? I think we were just touching the tip of the iceberg, and now we're starting to investigate that more. And I think it's important to know what these metabolites are, is because they might explain why some of my patients get side effects or not. And then also, what are these enzymes that are doing the metabolism? Because, if we don't want that metabolism to exist, then we need to know what those enzymes are so that we can target those enzymes. And so, these kind of sort of themes of research are part of my long-term goal to understand how the microbiome contributes to variation in patients and how we can better tailor therapy. And so I know I'm kind of running short on time, so I just wanted to say that, my take-home messages are that the microbiome varies among people, and this may contribute to patient disease and drug response, that microbes are likely affected by the drugs that we're giving to our patients, and the microbes also may act on the drugs that we give to our patients and can affect the ability of those drugs to be effective. And I think there's so much to learn. And this is my kind of plug to say that we need patients to enroll in these microbiome studies so we can learn more about the microbiome and modulate it, change it in a rational way. And so with that, I would like to thank you for your attention. Peter Turnbow was my mentor during this, but now I've actually started my own lab. And these are my collaborators, Jose Scher and Carla Subeda, and then the folks in the lab that helped me actually carry out this research. And then, of course, my funding sources, including the Arthritis Foundation and support from, the ANRF Arthritis National Research Foundation. That support from ANRF has been critical for me transitioning into my new role as like the head of my own lab. And I have been able to secure R01 equivalent funding. So I feel like I'm so excited to be able to do this, and support from the ANRF has been instrumental for that. And with that, I am happy to answer any questions.
Okay, Emily.
Yeah, yes, one question came in. I mean, is there anything patients can do to alter their microbiome to have a better, to see better efficacy with the drugs that they're taking? I know there's more work to be done, but as a patient at home, can they eat different foods or take different vitamins and stuff that would have a long-term or short-term effect?
Yeah, this is a question that always comes up in my clinic when I see my patients. And what we have a lot of data for. So there are a few things that really dramatically affect the microbiome, and antibiotics is one of them. I'm sure anyone who's taking antibiotics has experienced that phenomenon. And diet is another one. And we're starting to learn some of these other factors like drugs and how you were born. All those things are changing, but you can't change how you were born. You can change the drugs that you take, but you can definitely change the diet. And there's one kind of common theme that I've seen in the literature that if you're eating a Western or high-fat diet, that is generally worse for your arthritis. And usually it doesn't matter what your arthritis is. Like that high-fat diet or that Westernized diet is not good. So I can tell you what not to eat, but telling you what to eat is a totally different sort of guidance that I don't think we have the right guidance. And, there was actually a paper that came out from a group in Israel that showed that, if you have a banana and a cookie and like you give somebody a banana and you look at their sugar spike, or you give them a cookie and you look at their sugar spike in their blood, some people had a higher spike with the banana and other people had a higher spike in their sugar from the cookie. And that is just like, what? Like if you had asked a doctor which one's healthier for you in terms of, it would have been like, go with the banana. But like what that study showed is that the microbiome contributes to how you are digesting your food and how that's affecting your blood. So I guess that's a like kind of long-winded way of saying our microbiome shapes our response to diet. And it's hard then to say like one specific kind of diet can help each and every person. And so just like we're seeing kind of a precision medicine and a tailoring of medicine, I think we're also seeing that diet is the same, that we need to kind of tailor the diet to the person, because I think all of us have experienced this too, that we go on these diets because our friends have had amazing success. And then when we do it, we're like, what's going on here? And I really think that variation in the microbiome is contributing to that. So basically, the bottom line is, don't eat a Western diet, have a healthy diet this is what I tell my patients, have a healthy diet full of fruits and vegetables, and eat a diet that's good for your heart, like what Dr. Mclaughlin was saying, that's really important too. But then like the details of it are a bit harder to provide guidance on.
Absolutely.
Mm-hmm.
Thank you. Other questions for Dr. Nayak? Please type them into the Q&A.
Okay, so I will ask a question. So I'm interested in whether I realize there's only so many hours in the day. Is there evidence coming from anywhere that would point to other drugs that are typically used in rheumatoid arthritis interacting with the microbiome, or is methotrexate as a metabolically relevant drug somewhat unique as compared to a lot of the immunomodulators that are used afterwards?
Yeah, that is such an interesting question. So methotrexate, originally was designed to look like folic acid or folate, which is like a very essential vitamin that we tell pregnant women to eat all the time, right? And that actually we give to our RA patients when they're on methotrexate. So methotrexate is pretty unique because it was designed to look like a nutrient. Interestingly, like as I kind of did this research, I realized like some of our DMARDs are actually the same. Like azathioprine targets the same pathway. Mycophenolate also targets these pathways that are really essential for cellular growth. And so kind of the way I think about it, the two buckets are like the oral medications, like our DMARDs that are targeting kind of key cellular pathways. They are known, and there has been data from like in vitro, like studies in test tubes and petri dishes to show that the microbiome is affected by these oral medications, right? And they're oral, so like that interaction is relevant. But on the other hand, some of the biologics, the IL-17, the TNF, those were not designed to target pathways that are also in microbes. Those were designed to target cytokines produced by the immune system. And so I think of that as like a separate class. I don't know that anyone has tested those agents in a test tube. What people have seen is that patients that are on, for example, TNF inhibitor, once they start the TNF inhibitor, there's a change in the microbiome. And that's hard to kind of tease out, because then you don't know if the drug is acting on the immune system and that's causing a change in the microbiome, or if it's a direct effect of the drug. But yes, there is evidence to suggest that what I've shown you today for methotrexate is not unique to methotrexate, that it's also relevant with respect to other DMARDs that are commonly used in RA and other kinds of arthritides.
Fantastic.
Thank you.
Mm-hmm.
Okay, let me just see what time it is. It's a time that for us, I think it is time for us to go to the next one. Let me just take a really quick look at my sheet here.
Thank you, everybody.
Okay, 1 second. Oh yeah, okay. Well, so before we do, I wanted to thank you for a really great talk, and very clear and very interesting, and with a lot of practical implications, even if we're not ready to institute everything right away, but definitely with things that some of which can be instituted right away. So I really thank you for that. And I have to say, again, I won't tell you how old I am, that when I went to medical school, we just threw this whole microbiome into the garbage can, called it commensal, and said, you don't have to know about it. So be careful when somebody says to you, you don't have to know about that. I think there are a few things in the world that we really just don't have to know about. Okay. So we will go on to our final speaker for today, who is Anil Singh, who is an assistant research professor at the College of Pharmacy and Pharmaceutical Sciences at Washington State University. He is going to talk to us about IL-6 trans signaling that utilizes the ETS2 protein for synovial fibroblast invasiveness, or the generation of invasive synovial fibroblasts.
Dr. Singh, you might be on mute. We can see your screen, but we can't hear you yet.
Still the case. Sometimes you're not on mute, but your volume is way down. So make sure your volume's not way down.
Can you hear me?
Yes,
yeah, we can.
That did it.
Sorry about that.
No problem.
Thank you, Millie, for providing me opportunity to talk here, and I'm really thankful for funding me. And I've been working on rheumatoid arthritis for past 8 years or 9 years. Earlier, I was working on breast cancer research as a postdoc, first postdoc in Ohio State University before I moved to Washington State University. And since 8 or 9 years, my journey to rheumatoid arthritis is very exciting. Published several papers. And today I'm going to talk about, which I developed like over the span of time, like 8 or 9 years. My love is towards interleukin-6 signaling. But for the purpose of general audience, I'll start the heading as a title as Understanding Inflammation and Rheumatoid Arthritis. And did it go in the second slide?
Not yet.
Sorry, this system took over, like Zoom. Yeah.
Hey, James, was this the issue you were fixing before?
No, this looks like it's a PDF. Dr. Singh, are you able to hit that down arrow up at the top next to the number 1?
Oh, yeah.
There you go.
Oh,
Perfect.
yep, sorry.
No problem.
Because my control cursors are not working. I really don't know. So before I'll start, so rheumatoid arthritis is autoimmune disease, and it's mainly characterized by inflammation. And in autoimmune disease, everything is all about self-attack of your immune system. And there are various autoimmune diseases in the disease pathology. One of the best known is type 1 diabetes, and also lupus and rheumatoid arthritis. Overall, 5 to 10% world population is on the brink of autoimmune disease. And out of 5 to 10%, 1% is a rheumatoid arthritis contribution. And the only difference with the other autoimmune disease and rheumatoid arthritis is actually very localized to synovial area, which is joint. And also this particular disease happen 80% time to women, more than the reason is unknown. Like people say it's hormonal. People also talk about the genetic component. But as of now, hormonal part, genetic component part is all disputed. And rheumatoid arthritis start with the unknown trigger, inflammation. And also it start with a synovial seepage of blood cells, like T cells, B cells, another kind of cells. And it's trigger the environment inflammatory by producing more and more inflammatory cytokines, such as interleukin-1s, interleukin-6, interleukin-17, TNF-alpha. And they all act on synovial fibroblast. In rheumatoid arthritis, there are two major players, cellular player, because they are the resident cells. It's a synovial fibroblast, synovial microphage. And synovial fibroblast and synovial microphage is the one determines the inflammatory response more severe. And the fibroblasts contribute invasiveness, and they are very essential component of maintaining synovial integrity. And eventually they become panus-like cells. They like tumor, like big ball of cells, and they produce different cytokines, including IL-6 and also rankle. Rankle goes and crosstalk with osteoclast, and osteoclast chew the bone, and that's bone deformities, which we see in rheumatoid arthritis patients. So as I mentioned previously, this inflammatory condition is like a civil war where the cells are attacking on their own cells. And inflammatory cytokines such as IL-1, IL-1 alpha, IL-1 beta, TNF-alpha, IL-17, and IL-6, they all promote cells to differentiate, to become something different. And they're supposed not to be that kind of a cells. And ultimately leads to antigen presentation by some cells like a T cells, macrophage, and dendritic cells, and autoantibody production by B cells. And that autoantibody production to the B cells attack the self protein on different cells, like in this case, I would say fibroblast. And so in autoimmunity, it's all about the betrayal of self. So your cell makes several proteins. Some are known to the immune system all the time, but suddenly what if the protein has some mutation, like peptide mutation or the whole protein mutation? This all protein ultimately leads to presented by all the cells, which is nucleated cells, through the MHC complex, is also known as major histocompatibility complex. They present outside the cells and they says, hey, T cell and B cell, recognize, is this ours or someone foreign? And well, T cell comes and says, look like this is foreign, and let's trigger the inflammatory response. T cell present these cells to B cell, and B cell, without knowing, oh, this is self protein or mutant protein of your own protein, they trigger inflammatory condition worse and worse. And as I mentioned previously, that I works on interleukin signaling, interleukin 6 signaling. And so in the cellular system, interleukin 6 signaling is two types, classic and Tran signaling. So interleukin 6 signaling is not all the time bad. It's always needed for sometime pathogen invasion time when body heats up and clear the pathogen is also mediated by interleukin 6. So in classic signaling, what happened, every cell, most of the cell has a GP130 complex, also interleukin 6 receptor. It binds to interleukin 6, and upon binding, it triggers the JAK-STAT signaling downstream. And in Tran signaling, what happened is like interleukin 6 receptor is not there on the surface. Like say, for example, fibroblasts. Fibroblasts do not have interleukin 6 RS alpha, means interleukin 6 receptor alpha. And it comes from the blood. Like when infection happen, inflammation happen, liver produce a lot of interleukin 6 receptor. And ultimately, because it's floating in our blood, interleukin 6 receptor can bind to the floating interleukin 6, and it can just leach on GP130, and it can trigger a signaling. And that's, again, leads to JAK-STAT signaling. And there are two kind of a therapy on the JAK-STAT signaling mediated through the interleukin 6. One is a small molecule inhibitor, another is a biological treatment, such as tocilizumab, which inhibits interleukin 6 receptor and let the complex do not sit on the membrane of the cell, and the signaling will not even start. Other one is a tofacitinib, which binds to the JAK2, JAK1, and JAK3 complex, and it stops the signaling downstream. And as I mentioned, why interleukin 6 is so valuable. Interleukin 6, as I showed here, it's as a complex with an interleukin 6 receptor and GP130 complex. It triggers the signaling, and it has a pleiotropic effect. One, it can dictate B cell to produce antibody, keratinocyte site to proliferate, mesenchymal cells to proliferate, T cell, it can regulate T cell, and also a platelet production, and also it can promote the growth of various cells. And also it can promote the hematopoietic stem cells to promote in combination of VEGF. It's another cytokine, and it creates angiogenesis. So it's pretty much like a cancer-like microenvironment. And in combination of Frankel, it promotes the osteoclastic differentiation. And as I mentioned, osteoclastic differentiation is one of the main causes for bone deformities in RA. And here comes my work related to osteoclastic invasion. So one of the things which I did to start with this work is funded through ANRF and recently published 6 months before in Frontier Immunology. And what I found when you grow this synovial fibroblast for a long time, like 12 days to I went to up to 21 days actually. So what it do, it start chewing the bone, osteogenic surface. It's a very fine coat of the bone, and it's start creating a hole. And we stain this osteogenic surface through the toluidine blue, and we realize that if you use, there is one cytokine over here. It's also known as MCSF Rankel. They are osteoclast promoting cytokine. I can use as a combination and also alone. And I realized that IL-6 trans signaling can induce osteoclast-like phenotype in synovial fibroblast. And I realized it's not a good thing, because synovial fibroblast is supposed to be structural cells which contribute many other roles. And its invasiveness becoming to the osteoclast is not a good sign. So I also characterize some other protein which is present in osteoclast, such as TRAP. TRAP is a also known as tartrate-resistant acid phosphatase. It's a very unique to osteoclast. And I realize only IL-6 plus IL-6 combination produces a lot of trap from the cells. It's chronic exposure leads to the overproduction of trap. And when we check the secretome of this synovial fibroblast, there is 113 unique protein contributed by synovial fibroblast upon IL-6 trans signaling. And among these unique proteins are if you do a gene ontology study from the 113 protein, it characterizes that it's actually enhanced the metabolism of cells, like glycolysis and phagocytosis. And also uniquely, it's differentiating as a stem cell differentiation parameters, like over here. So IL-6 upon for 12 days or chronic exposure, it's actually transformed the cells to a different cell. It's not the same cell which started with. And I recently found out a transcription factor, H2. It's a part of, it's a nuclear protein, and it binds to the DNA and regulates the transcription of many other proteins in the cell. And it helps. And when you reduce when you knock down or remove this protein H2, this trap positive cells disappeared. And look like it's a promising that rheumatoid arthritis fibroblast, which become bad in response to interleukin-6 signaling, H2 can take care of many things, such as downregulation of cathepsins, which is also characteristics of osteoclast, cathepsin B, cathepsin K, also marker for invasiveness, which is podoplerin. And this finding is very unique. And we are also expanding this work towards different grant. And right now I'm second year funded, so I'm putting more and more resource to the sequencing, chip sequencing of this transcription factor. Let's see what this H2 gives the different unique or insight. And potentially in future, I'll be able to find some therapeutic advantage for H2. And also, I would end up with the acknowledgement slide. Dr. Salahuddin Ahmed is my mentor and collaborator. And also David Fox from University of Michigan, he's a collaborator. And my friend Madhu, who helps me in pathology study, funding for ANRF, and also Dr. Ahmed funding for NIH. I can take the questions.
Emily?
Yeah, thank you so much, Dr. Singh. So obviously you talked a little bit about ETS2. Do you think that finding more information out about that and how it relates to IL-6 will improve the efficacy of some of the IL-6 drugs, or maybe lead to new drugs for patients?
So finding about ETS2, it's all about how ETS2 reprogram the cells. Because I saw it's metabolically engineered cells to some different kind of cells, differentiate to the stem cell-like phenotype, and how it's to shut down everything, like invasiveness of fibroblast, and also osteoclastic nature of fibroblast, which is a unique. And so it's a promising, it's to know, what could be a better therapeutic target. Could be possibly some drug inhibit H2, because H2, I got this result because of inhibition of H2. So possibly IL-6 utilizes H2 for its bad role.
Okay. Wonderful. Any other questions for Dr. Singh today, or any of the other panelists?
Well, I see we're really out of time, but I just would ask, I know that there's been some effort actually to develop ETS2 inhibitors. Do you know if that's made any progress?
No, not much, because the S2 is not fully crystallized. And there is people talk about targeting through the monoclonals, but this is transcription factor. It has to be targeting inside the nucleus. So the small molecule inhibitor is the best way. And only pointed domain and DNA binding domain is crystallized. And with my collaborator, he's checking all this, and he's not able to very well establish if we can inhibit, based on the crystal structure which we have. And I really wanted to crystallize this protein, but it's actually very ambitious. And maybe I will propose in RO1s in future.
Okay. Well, I have a few more, but I'll send them to you through the ANRF site. And I just want to thank you for a very informative, interesting talk. The idea of a transdifferentiation from a fibroblast to an osteoclast, I think, is very intriguing. I'd be really interested to understand more about the mechanism of that and whether there are other triggers for fibroblasts, synovial fibroblasts becoming osteoclasts. But again, as I said, I'll send you those questions later.
Please.
And thank you again for joining us today and giving a lovely talk.
Welcome.
Yes, and thank you to all of our presenters and to Dr. Mellins today for moderating. It's been a credible array of presentations from our scholars, and I think you all can see firsthand the impact that ANRF has had in supporting these incredible individuals' quest for a cure. I would like to thank once again our sponsor, AbbVie, for their support. And lastly, thank you to you, everyone who joined us for this great event. If you enjoyed this webinar, we kindly ask that you consider making a donation today by visiting curearthritis.org/donate. Every contribution helps us to achieve our mission. And please remember to follow us on social media as well to stay connected. We look forward to seeing you next week at the next webinar. Thank you.